The Antibacterial Properties of Honey by Lindsay Conrad. What is honey? Honey is a thick golden liquid produced by bees by using the nectar of flowering plants. It's composed mostly of sugar and water and also has small amounts of minerals and vitamins as well as microbes. Honey is described as a reservoir for microbes due to its association with pollen, the digestive tract of honeybees, dirt, dust, air, and flowers. A brief history of honey. Ancient Egypt. Honey was the most popular Egyptian drug, being mentioned 500 times in 900 remedies. Honey was utilized for its antibacterial properties that helped heal infected wounds. Almost all Egyptian medicines contained honey together with wine and milk. They also used honey for embalming the dead. According to the text of Ayurveda, honey was used for those with weak digestion. It's emphasized in the text that the use of honey is highly beneficial in the treatment of irritating cough and valuable in keeping the teeth and gums healthy. Additionally, traditional Ayurvedic experts recommend honey for skin disorders such as wounds and burns. Hippocrates, the great Greek scientist, utilized honey for baldness, contraception, wound healing, laxative action, cough and sore throat, eye diseases, topical antisepsis, and prevention and treatment of scars. What do these all have in common? That they all used honey for burns, skin wounds, and sore throats. The main reason why honey has antibacterial properties is due to the amount of hydrogen peroxide present. Hydrogen peroxide works to oxidize surrounding cells. The more hydrogen peroxide available, the more potent the honey is as an antibacterial agent due to its ability to cause significant cell damage. The amount of hydrogen peroxide naturally present is directly dependent on the amount of the enzyme present, glucose oxidase, which converts glucose to gluconic acid and hydrogen peroxide. Other reasons why honey has antibacterial properties are due to its non-peroxide components such as phytochemical factors, which are believed to be many complex phenols and organic acids, often referred to as flavonoids. These complex chemicals do not break down under heat or light or are affected by honey's dilution. The most direct evidence for the existence of non-peroxide antibacterial factors in honey is seen in the reports of activity persisting in honeys treated with catalase to remove the hydrogen peroxide activity. Honey is a supersaturated solution of sugars, 84% being a mixture of fructose and glucose. The strong interaction of these sugar molecules will leave very few of the water molecules available for microorganisms. Honey is characteristically acidic with a pH of between 3.2 and 4.5, which is low enough to be inhibitory to many animal pathogens. For example, the minimum pH values for growth of some common pathogenic species are E. coli 4.3, Salmonella species 4, Pseudomonas aeruginosa 4.4, and Streptococcus pyogenes 4.5. Thus, in undiluted honey, the acidity is a significant antibacterial factor. Staphylococcus aureus is a gram-positive bacteria. It's pathogenic, causing infections in immune-compromised people. Some strains, such as MRSA, have developed resistance to many antibiotics, making them tougher to treat. However, honey has shown effectiveness on antimicrobial resistant strains, including MRSA. Honey has also been shown to inhibit biofilm formation of Staphylococcus aureus. This table shows the results from an experiment done by Ronnie et al., which was published in the Journal of Clinical and Diagnostic Research. It shows the antimicrobial sensitivity pattern of methicillin-resistant Staphylococcus aureus and methicillin-sensitive Staphylococcus aureus with reference to honey and other common antimicrobials. The results show that methicillin-resistant Staphylococcus aureus was susceptible to honey by displaying a large diameter of zone of inhibition of 36.2 millimeters, compared to the zone of inhibition from the other common antimicrobials. MRSA was still resistant to all of the other antimicrobials except for linezolid. As for methicillin-susceptible Staphylococcus aureus, honey was either more or just as effective as the other common antimicrobials used. From the same study, we could see that honey was the most effective antibacterial agent against MRSA, methicillin-resistant Staphylococcus aureus. This picture shows the results from our lab in which a TSA plate inoculated with Staphylococcus aureus showed that it was susceptible to just regular honey. This honey that I used was from a packet from a cafeteria, so it wasn't medical grade at all. And I dipped a disc in the honey, put it in the middle of the plate inoculated with Staphylococcus aureus. After 3 weeks, a zone of inhibition was seen, which was actually quite large, showing that this Staphylococcus aureus strain was susceptible to just regular honey, not medical grade at all. Pseudomonas aeruginosa is a gram-negative bacteria that's commonly found in hospitals. It's an opportunistic pathogen that loves to thrive and grow in moist environments. Pseudomonas aeruginosa has become multi-drug resistant due to its potential to acquire new antimicrobial resistance. Its activity is enhanced by its ability to form biofilm. In a study where 50 strains of Pseudomonas aeruginosa isolated from infected wounds were evaluated for their antibacterial action using honey in comparison with different antibiotics, the results showed that all strains were found to be sensitive to honey at a minimum inhibitory concentration of 20%. Also, all the isolates of Pseudomonas aeruginosa tested were killed in 12 to 24 hours, depending on the dilutions of the honey tested. These results prove that honey could prevent the growth of Pseudomonas even if it was diluted by deionized water. One of the promising aspects about using honey as an antibacterial agent is that the composition of honey is extremely variable and reflects the environment in which it was created. This is important because bacterial resistance isn't as likely to occur if each type of honey has its own specific composition. Currently, it's believed that Manuka honey from New Zealand and Australia is the most effective type of honey for killing bacteria. Manuka honey has very high levels of methyl glyoxal, which is one of the non-peroxide components found in its specific composition. The amount of methyl glyoxal in Manuka honey is displayed on Manuka honey label as the UMF, which stands for unique Manuka factor, and that goes from about +10, +15, +20, and the higher the UMF is, the more effective that honey will be in killing bacteria. So why is any of this important? It's important because we need alternative antimicrobial strategies urgently due to the increased frequency of resistant pathogens that are developing and spreading globally. These resistant pathogens that are spreading globally are decreasing the effectiveness of the antibiotics that we currently use as a last resort. To sum it up, honey has been used throughout time as a therapeutic type of medicine for skin disorders, colds, sore throats, burns, etc. It's only now being rediscovered in Western medicine due to the increased resistance of pathogens spreading worldwide. And it's also interesting to note that future research is needed to compare the effectiveness of honey used in combination with other antimicrobial agents.